That is a tough act to follow. Really, thank you so much for sharing your story. That was really amazing. So there's a lot of opportunity to do some great science, and we're going to hear 3 wonderful talks in this first session on next generation screening technologies at scale. Our first speaker is Gabe Griffin. Gabe is a physician scientist at Dana-Farber and a member of the Broad Epigenomics program, and he's going to talk to us about his work on cancer immunotherapy and understanding how to make it work better. Gabe, take it away.
Last talk. There's a lot of really important work that we can do in the rare disease area. The particular topic that I'm going to speak to you about today is a collaborative project we're doing. It is attempting to harness functional genomics and machine learning for rare cancer research, and particularly to find new drug targets for rare cancers. And I'll just say at the outset, this project is a collaboration with the Iqbal Lab here at the Broad Institute. The Iqbal Lab is run by Dr. Sumaya Iqbal, shown at the top here. And a lot of the work that I'll speak about was also led by Dr. Lauren Wickstrom, who I believe is here as well. Okay, so the talk is organized into 3 categories. Our goal is really to find new drug targets for rare cancers, as I said. So the first part will be why rare cancers? Why should we care about rare cancers? The second part is what are the challenges of drug discovery in this area? And then the third is what is at least our proposed solution to these challenges? What is the strategy we're taking? Okay, so for the first point, why rare cancers? So if you'll bear with me for a moment, I'd like a show of hands from the audience of anyone who has been either personally impacted by cancer or had someone in their immediate sort of world impacted by cancer, whether it's a family member or a friend or a coworker. Okay, so it's a fair number of people. You can keep your hands up if you don't mind. And so I'm going to cut this group in half. So anyone who was born on an odd numbered day, you can lower your hand. So if you were born on an even numbered day, you can keep it up. Okay, great. And I'm going to cut that group in half one more time. So anyone who was born from January through June, you can lower your hand. And if you were born July through December, you can keep your hand up. Okay, so there's maybe 10, 15 hands still raised in the audience. So what am I getting at with this? The point is that among all cancers, okay, while any individual rare cancer might be rare, among all cancers, rare cancers come together to comprise about 25%, right? So about 25% of everyone who raised their hand initially has been in some way affected by a rare cancer, okay? So they impact all of us. The formal definition of a rare cancer maybe isn't that important, but it generally means cancers that affect about 40,000 or less new people per year, or 15 per 100,000 cases per year. What is the challenge of studying rare cancers? Well, fewer patients means it's harder to test possible therapies. It's harder to get samples for research. It's harder to get funding sometimes. It's harder to make progress. And this problem is particularly acute in the field of rare pediatric cancers. So in effect, every pediatric cancer based on its incidence is considered a rare cancer. And so you can see the numbers in terms of cancer diagnoses and cancer deaths on an annual basis in individuals less than the age of 19 here. But these are all kind of statistics, and I think especially following the last talk, I think maybe one of the most critical things to remember, especially from sort of a physician-scientist perspective or perhaps from a patient perspective, is that cancer always affects an individual person with a family, and they don't choose to have a rare cancer, they just have their cancer. And so I think it's important that we take these cancers seriously for study and therapeutic development like we would anything else. So I think that kind of relationship of an individual patient is highlighted in this statue here. This is actually a statue of Dr. Sidney Farber, who helped start the Dana-Farber Cancer Institute. And one of his early patients, who was nicknamed Jimmy, he was a boy who was diagnosed with a rare form of blood cancer. This statue actually sits right outside the laboratory building, the Jimmy Fund building where our lab is. And so, walk past this statue essentially every day on the walk in from the parking garage. And so it really serves as a source of inspiration. One of the key discoveries that Dr. Farber made, this is back in the 40s, was that cancer cells depend on this nutrient folic acid for their growth. And if you could use a drug that targeted that pathway, anti-folate drugs, at the time it was called aminopterin, you could actually kill their cancer cells, in this case blood cancer. And aminopterin is really the predecessor of this drug that's widely used now called methotrexate. And in this remarkable story, Jimmy was treated with this drug when he was a kid. He was cured, and he lived to the ripe old age of being a grandfather. And there's this beautiful photo from 1998 of him with his grandkids. His real name is Einar Gustafsson. He was originally from Maine. So I think really transformative potential for individual people for cancer research and particularly in rare cancers. Okay, so what are the challenges of drug discovery in this area? How can we make progress similar to what I just showed? So I think in kind of the early days of cancer research and drug development, there was this sentiment or perhaps a hope that cancer would be a single disease and that we would find a single magic bullet that would work for, if not all patients, most patients. So that was really a dominant aspiration or hope in the field. But as we've realized in decades since, cancer is in fact many, many different diseases. This is one of the challenges of developing cancer therapies. You might find something that works very, very well for a particular disease, but it won't work at all for something else. So really what we need is many, many different magic bullets. Some people have even described like a magic shotgun approach where you really hit an individual patient's cancer with at least one or several targeted therapies. To highlight this development, so in Jimmy's time, there were two major types of acute leukemia, or this acute form of blood cancer. They were broadly categorized into acute leukemias that had differentiation like myeloid cells or differentiation like lymphoid cells. There were basically these two big categories. And this is a field that's near and dear to my heart. In my clinical life, I help diagnose patients with blood cancer. And this field has evolved tremendously. This is not meant to see the details here, but this is just meant to highlight all the many different categories of acute leukemia that exist today. There's B acute leukemia, T-cell acute leukemia, myeloid leukemia, and they're all defined largely by very distinct genetic drivers, many of which are oncogenic gene fusions, and we'll talk more about that. So there's just incredible diversity of drivers within even just this one disease. Luckily, there has been, despite this diversity, enormous success in the development of targeted therapies for many of these diseases over the years. Highlighting just a couple here, one example is chronic myeloid leukemia, or CML. It's driven by this oncogenic driver BCR-ABL. There was a tyrosine kinase inhibitor, imatinib or Gleevec, that was developed for this, FDA approved in 2001, and this has basically transformed therapy for these patients. A couple other examples here targeting other particular fusions in blood cancer that have been developed. Now, one thing you'll notice about these is that these are all, the mechanism of action of these drugs are all tyrosine kinase inhibitors. What that means is that these drugs are targeting an active site, an enzymatic site, a kind of conventional druggable site on these oncogenic fusions. That's one of the reasons these have been possible. So why not just use this strategy for all other oncogenic fusions, and we can cure all rare cancers? Well, the problem with that is that many key drivers of rare cancers and many oncogenic drivers or oncogenic gene fusions actually lack conventional druggable sites. They lack enzymatic domains. And so they're kind of less straightforward from a drug development standpoint. You don't necessarily know where to design your drug against. Here's one example of that. This is an example of an MLL or KMT2A gene fusion that drives certain subtypes of acute leukemia. On the top is what the wild type protein looks like. So this is a lysine methyltransferase in its sort of wild type physiologic form. And the enzymatic domain, the histone methyltransferase domain, is shown all the way out here at the C terminus, this set domain. In the fusion protein, the N-terminus of wild-type MLL is retained, and then there's a break, and one of various gene partners can come on the C-terminus. And this essentially causes the loss of this enzymatic domain. So this kind of conventional approach, enzymatic targeting, disrupting an active site, won't work for an MLL fusion protein. Okay, so what can we do? How can we go after this? Well, MLL is actually a beautiful example, in large part due to work that has been done locally. There's been enormous therapeutic progress in this area, and a key insight was the discovery that MLL fusion proteins actually form oncogenic protein complexes, and that's illustrated here. So you can almost imagine the fusion protein acting as a scaffold for recruiting all these other transcriptional regulators, including this protein menin, and that oncogenic transcription complex finds target genes that are critical for promoting the leukemia. So if somehow you could disrupt this complex, you would prevent activation of these leukemogenic genes and potentially cure patients' leukemia. So that has been an enormous success story. So there's an inhibitor, actually several different inhibitors of this protein menin, which these drugs in general disrupt protein-protein interaction between menin and this oncogenic fusion, the MLL fusion, and disrupts its ability to activate leukemogenic genes and is very effective in MLL rearranged leukemia. So I think there's a nice paradigm here for thinking about how to drug oncogenic fusion complexes in a way that doesn't require, kind of a conventional enzymatic inhibition approach. And of note, this MLL inhibitor, revumenib, received FDA approval recently in 2024. Okay, so how can we build on this kind of paradigm of menin inhibitors for MLL rearranged leukemias and extend it to other leukemias and other oncogenic fusion-driven rare cancers? That was really the origin of our work and collaboration. This is just a summary. I won't go through all the text, but basically the targets that we're interested in are oncogenic fusion proteins in rare cancers. Our hypothesis is that the oncogenic fusion proteins form these complexes that are critical to cancer growth. Additional hypothesis is that if you can disrupt those complexes, these will be targetable, effective dependencies for rare cancers. And the objectives of our work is really to define the functional interactors of oncogenic fusion proteins in rare cancers, but to do this in a way that is really scalable and cost efficient. So perhaps to accelerate this process beyond kind of conventional approaches. And to do that, our strategy is to integrate some of the high throughput technologies, scalable technologies that have been developed over the last decade and beyond. First in functional genomics, that's what my lab really specializes in, doing CRISPR screens to identify new therapeutic targets. And then also structural modeling of oncogenic fusion proteins using some of these just revolutionary computational structural biology tools that have come out, including things like AlphaFold. Putting these two together, we want to prioritize new drug targets for oncofusion positive rare cancers. Okay, so in the subsequent slides, just have a few more slides to show our progress thus far in this project. I'm going to be using the MLL oncogenic fusion in leukemia as kind of a benchmarking control for our method development and for illustration purposes. Okay, so this step one of our method is to identify cell lines from the cancer dependency map that was generated here at the Broad Institute that have a functional dependency on a given oncogenic gene fusion. So in this particular case, we're looking at all of the DEP map data. Each dot here is a cell line. Okay, and then we're looking on the x-axis of the effect of knocking out one component of this gene fusion, MLL or KMT2A. And you can see that cell lines to the left, these are cell lines that die when you knock out that oncogenic gene fusion. These indeed are highly enriched for leukemia cells that harbor this oncogenic gene fusion. So it's a way essentially of identifying cell lines that have a strong functional dependency on this oncogenic gene fusion. So we collect all those. Next, we then identify among those cell lines selective gene knockout dependencies that are really unique to that particular oncogenic gene fusion. In other words, these are genes that if you knock, like other genes, that if you knock them out in that cell line, the cancer cells will die as well. And so that this is a group of those that come out of this kind of analysis. And what you can see here is that this approach, in a kind of unbiased way, also finds menin, this known interactor of MLL oncogenic fusion, and a known drug target in this context. So the hypothesis would be that there's probably other members of an MLL fusion protein complex that are included in these codependencies. So the question then becomes, well, how, given this list of codependencies, say we took the top 50 from this, how can we then associate them together? Are they all just random genes, or do they actually come together to form true fusion protein complexes? So essentially, how could we group them into little subgroups? So one strategy is to use an approach like string databases. These are beautiful databases that have been kind of built on the top of all of the literature and published data that's come out over the decades. A lot of it experimentally informed, where people have pulled down particular proteins and done things like mass spec to find what the protein interaction partners are. This is a great starting point, and you can see that even in string you can find interaction networks that involve menin, MLL, and some of the known oncogenic fusion partners. So this is one starting point for doing this. But this method is also somewhat limited by the fact that for this database is populated by known information or published information. So it would also be nice to complement this with a way that sort of a strategy that can associate proteins without any reliance on published data, or perhaps you in a context where maybe a protein hasn't been studied before. How could you approach that? And this is where AlphaFold comes in, and this has been the work largely led by the Iqbal lab. So you can use, in short, AlphaFold can allow you to predict the structure of a protein just from its primary protein sequence. And there are newer versions of AlphaFold that can also allow you to do protein-protein interaction modeling to potentially identify protein-protein interaction surfaces that could be druggable. So we took all of those codependencies, and then without any prior knowledge about them other than what AlphaFold could teach us, we tried to do pairwise interactions between all of them. And what's shown here is the result of that. So if you do pairwise interactions between all of these individual codependencies and the MLL fusion protein, this is what you see. Kind of gratifyingly, menin, so this established target, is at the top of this list. It's the fourth highest sort of confidence predicted interaction with the MLL oncogenic fusion protein. And so this really gives us confidence that this type of strategy, if applied to other oncogenic fusions, could identify meaningful PPIs. And just to drill down on this a little bit further, this is this beautiful representation where there's the menin protein in tan, and there's this menin interacting peptide of MLL, which is in aqua, and then a bunch of the individual contact points between these two that were identified as of high confidence by AlphaFold. And so it can really help you even get down to the molecular level or structural biology level to find interaction surfaces that you could then exploit for drug development. Okay, and then the last step, once we have these structural computational models with all these PPI surfaces predicted, then the question becomes, well, how can you validate their function? And this is where we bring back in or circle back to the functional genomics approaches, integrating base editor tiling mutagenesis of oncogenic fusions, where we're basically introducing point mutations across the entirety of a fusion and testing the functional effect of that in high throughput screens. And once we have that data, we can then integrate that with the structural predictions to nominate candidate drug binding sites. And so this is an example of doing this for the menin protein, where this is actually reanalysis of published tiling data from this reference for the menin protein. And you can see that there are a number of resistant hits that come out of this screen. These are basically mutations that prevent menin inhibitor effects in these cells. And then you can then plot those resistant sites onto these structural predictions that we've generated, and find that indeed these sites affect known drug binding sites and kind of make sense from a structural standpoint. Okay, so the next steps for this is to develop this general approach now that it seems to be working in this positive control or benchmarking context, develop it as a scalable workflow to identify novel interaction sites in additional or potentially all oncogenic fusion positive cancers that we have some functional dependency data for. And we're specifically focusing our efforts now on leukemia or blood cancers, and also increasingly now in pediatric sarcoma. Okay, so with that, I'll stop. Acknowledgements to folks in my lab, Jada Coffey and Annie Chen, and then also in the Iqbal lab, Dr. Lauren Wickstrom. And also thank you to Guillaume Ponce-Montagne from Broad CDOT, who's been an excellent structural biology collaborator, and also to the Ladders to Cures Accelerator for pilot funding for this project. Thank you very much.
Okay, as Jillian mentioned, we're going to do a Q&A after all three speakers. So I'd like to welcome up our next speaker, JT Neal. JT Neal is an institute scientist here at the Broad. He is the director of systems genomics in the metabolism program and part of the Novo Nordisk Center. And yes, I just memorized that right now. JT, take it away.
Perfect. All right. I'm really excited to be here representing our team. I'm going to actually stand out here because they don't build these podiums for people of my stature. Our project is called Rapidly Identifying Drugs for Rare Disorders by Pooled Image-Based Profiling. I'll be giving the talk today, but I actually am co-leading this project with Ann Carpenter, who's here at the Broad. So I don't think I need to convince really anyone in this room that therapeutics are a major unmet need for rare diseases. And as you all are aware, rare diseases collectively are not rare. As many as 30 million Americans, or nearly 10% of people in the US, are affected by a rare disease. And they cause over a third of deaths in the first year of life. So this is really a heartbreaking medical problem. Additionally, there are thousands of these diseases, perhaps as many as 10,000. And there are approved treatments available for 5% or fewer of these. So this is really a huge clinical gap that needs to be filled. And part of the reason this gap exists is because drug development, canonical drug development, de novo drug development, is both incredibly expensive and incredibly slow. So we can take anywhere from 10 to 17 years to develop these agents from scratch, and it can cost over $1 billion. Now, this makes rare diseases not a particularly good commercial candidate, because unless we can figure out a way to aggregate patient populations, there's no way for drug companies to recoup this money. Plus, people are suffering from these diseases all the time, and we just don't have the time. So we and others, actually many others in the rare disease community, have come to rare disease drug repurposing as a promising approach to treat these disorders. So in drug repurposing, we are identifying new therapeutic uses for drugs that have already gone through some level of testing. It takes advantage of some of these early pipeline steps that fail out many early phase compounds. So things like solubility, stability, selectivity, and potency. But most crucially, these drugs have already been proven to be safe in humans. And so this means that the timeline to get this drug into a patient can often be much, much faster, and at a pretty dramatically lower cost. Well, if you're interested in doing drug repurposing, the Broad is really a wonderful place to be, because we have one of the world's best collections of drug repurposing compounds in a resource called the Drug Repurposing Hub. The Drug Repurposing Hub is a very, very large collection of compounds, over 7,000, that sort of span a range of clinical phases, and a large number of targets and mechanisms of action. And importantly, it has over 2,400 FDA-approved drugs. So a really, really great resource, first in class for someone who wants to run screens trying to identify drugs to repurpose for rare disease. But we're still facing a pretty big challenge, and that is that these rare diseases are incredibly diverse. So there's something like 5,000 rare Mendelian diseases where we know a genetic cause. We have a genetic anchor, and thousands more where we don't. And these diseases affect dozens of unique cell types and organ systems. And so, as Anna alluded to this morning, many of us in the community are trying to find these nodes or these handles by which we can reduce the complexity of this problem. So can we identify some sort of common mechanisms that allow us to have an entry point? And it turns out, actually, that protein mislocalization is a mechanism that's common to many rare diseases. So I'm showing you here a figure from an elegant study from Anne Carpenter here at the Broad and Mikko Taipala at the University of Toronto, in which they screened a very large number of rare coding variants. And so they overexpress these as proteins, and what they observed is that these, and they're tagged with flag tags, so you can see where they go. And what they observed is that many of these proteins are mislocalized, so they end up in the wrong part of the cell. So for instance, if it's supposed to be in the Golgi, it ends up in the ER or vice versa. This happens often along sort of known secretory route within the cell. And that these things also tend to fall in sort of patterns of mislocalization shared between, some of these protein variants. And it's not all that uncommon. 16% of all the variants they test have one of these mislocalization defects. And intriguingly, the degree of mislocalization is often associated with the disease severity. So the amount of protein that's in the wrong place can lead to more severe symptoms and earlier onset of disease. So this leads to a really tantalizing question. What would happen if we could move some of this protein back where it's supposed to be? Well, it turns out that we actually don't have to stretch our imagination all that far because correcting protein localization is a proven therapeutic strategy. So probably the best example of this is in cystic fibrosis. So the CFTR protein, chloride ion channel that's supposed to be at the membrane. In cystic fibrosis-inducing mutations like CFTR delta 508, you see here we have diffuse staining. But it turns out that if you treat with Vertex's drug, lumacaftor, you can actually drive some of this protein back into the membrane. And when you then combine this therapy with ivacaftor, which enhances activity of this channel, you can actually achieve significant therapeutic benefit. And this has been observed for other cases. Here I'm showing you progeria and lamin A, which I think many of you are probably familiar with, in treatment with the agent lonafarnib. So our goal really is a pretty simple one, and that is, can we identify new drug repurposing candidates capable of correcting protein mislocalization in rare disease? Pretty straightforward. So step one, identify the pathogenic phenotype. So here we have a very clear screenable phenotype. That is, the protein is in the wrong place. We are combining that with a little bit more complex phenotype, so cell profiling with cell painting. And then we just want to test drugs, screen drugs to see if we can reverse this phenotype. So can we make unhealthy cells look more like healthy cells? So this is pretty simple, as I said. Why has this not been done already? Well, practically speaking, it's actually quite difficult using traditional screening methods because there are, if for instance you wanted to screen all 7,000 drugs in the repurposing library against just our 250 variants, you're talking about 1.75 million individual experiments plus controls and replicates. A measly 600,000 if you just want to do your FDA-approved drugs. And we're not afraid of scale here at the Broad, but this gives us pause. And this is because we're screening these phenotypes optically. And that means that we have no way of taking these variants and pooling them together, because then we wouldn't know which cell had which one. Thankfully, we're not held back by this any further because of a technique developed in Paul Blaney's lab called optical pooled screening a few years ago. What this enables us to do is to put unique barcodes onto each of our disease variants, amplify these barcodes, and then sequence them directly on a microscope using Illumina chemistry. You'll hear more about this in Matt Brown's talk after mine. Here's what it looks like. So we can sequence on a microscope over sequential cycles. Each one of these dots is a base in the barcode. And so through sequential cycles, we can figure out what the barcode is, what cell has what variant. This now enables us to put them all together into one big experiment. And we've spent the last few years, my lab working with Paul's lab, Anne's lab, and some collaborators at Calico to really platformize this. So this is the essence of our L2C project. We want to take our 250 disease variants along with 100 of their wild type partners, pool them all together, and apply a compound from the drug repurposing library. We'll collect variant localization and cell morphology, and combine that with optical variant barcoding to enable us to assess compound effect and assign a variant to cell. So this is a huge increase in throughput. So here for screening 100 drugs, if you did it the old-fashioned way, it would take 35,000 experiments. We can do it in 100 experiments. So our aims were to build and QC a bar-coded library of these 250 disease variants and their wild-type controls, capture protein localization and paired cell morphology using cell painting for both the normal and the disease-associated variants, and then to screen 100 FDA-approved drugs against pooled variants to identify potential therapies. We're making good progress to date in the first year of our project. So we've finished building the library and we've QC'd it. We've optimized all of our screening culture conditions, staining, in situ sequencing. We've carried out a pilot cell coverage study to let us know how many times we need to see a variant in a cell to be able to identify hits. And actually, as we're here in this room together, we're carrying out a pilot screen in which we're screening 4 drugs from the repurposing collection against our 100 disease collection. We hope to read that out next week. So of course, it's worth spending a minute to ask how we're going to think about hits that come out of the screen. We have no illusions that we're going to take a primary screening drug and put it into a patient. So we're screening these all in singlet to begin with, 1 high dose and 1 well. These are pooled experiments, and so we think this helps alleviate some of the batch effects you would normally see in arrayed drug screening. But any hits we find, we're going to take back and reconfirm in 8 replicates across 8 doses to build a dose-response curve. Any hits from the secondary screen we'll take into a tertiary screen that will be in a cell type. So we're screening only in a single cell type, I should have said, Helichiodo cells. We will then take those compounds and screen them against that variant pair now in a disease-relevant cell line. So be it neurons or endothelial cells, whatever is appropriate for that condition. And then we're going to take this back to the rare disease community. We're going to bring it back to the Ladders to Cure community, and we're going to disseminate these data through traditional venues, publications, and online portals and the like, work with gap-bridging organizations like NCATS and Every Cure, and then engage other rare disease researchers to see if we can motivate further experiments, be they in vitro or in vivo, as well as engaging clinicians where we have promising candidates to see if we can move into clinical trials or potentially even consider compassionate use exemptions for cases where there is great evidence and extreme need. And then we hope, of course, that through screening these multiple mechanisms, we'll be able to identify shared nodes that might allow us to aggregate diseases into shared classes, which might make them more interesting candidates for drug companies to want to develop drugs for. And before I wrap up, I want to take a moment to especially thank Anna and Jillian and the rare disease community and Ladders to Cures here at Broad, because this work is really, really hard to get funded. And we have been trying to get this project funded, especially Anne has been trying to get this project funded for years. And the pilot funding that we're using here through Ladders to Cures as one of the initial projects is enabling us to generate not only key proof of concept data, but has given us momentum. And that momentum we have actually used now. Anne and I are in the early stages of forming a industry collaboration, a consortium, the VISTA consortium, that will begin the first process of scaling this effort now from 100 drugs to 500 drugs. And so this is not finalized. Keep your ears open. But we have 2 pharma partners already that are very, very excited about what this data set could mean. And then, of course, in the fullness of time, this genetic discovery is ongoing, and large projects, including ones like the one that Anne is currently leading, are going to discover more and more of these cases. And so in the fullness of time, of course, we have the bold vision of being able to screen all of the Broad drug repurposing library against thousands of rare diseases to find new medicines for the patients who need them most. So with that, I'll thank the people who've done the work in my lab, in Mikko Topali's lab at the University of Toronto, especially my partner in crime, Ann Carpenter, and the Broad Imaging Platform, and Paul Blaney's lab. And then obviously huge thanks to the Ladders to Cures Scientific Accelerator for making this possible.
Hey, thank you so much, JT. Our final speaker of this session is Matt Brown. Matt received his PhD from Mayo Clinic. He then came to Anna Greca's group to do his postdoctoral work, where he's also an awardee of the Broad Ignite Foundation. So Matt, take it away.
Thank you. Thank you, John, for the really nice introduction. In the interest of time, I'll try to get everyone to the coffee break as soon as possible. But I'm really excited to share our work today about using multiplexed optical pooled screening for proteinopathy therapeutic discovery. So for a long time, I've been interested in how the kidney works, and specifically how kidney epithelial cells, which line the nephron or the kidney's functional unit, function to transport solutes and waters across the apical and basolateral membranes of the kidney. These epithelial cells do this by moving transporters and other accessory proteins to these membranes, and they do this through a pathway called the secretory pathway. In these cells, these transporters need to be synthesized in the ER, processed in the Golgi, and then moved to wherever they need to function within the cell. And then eventually they're degraded using lysosomal or proteasomal mechanisms. However, mutations in protein cargoes, such as MUC1 or uromodulin, or disruption of the secretory pathway itself, can lead to proteinopathies and a number of rare diseases in the kidney and in a number of different organs. And previous work from our lab has identified dysregulation in a kind of a nodal mechanism called these TMED cargo receptors. And these TMED cargo receptors live in the space between the ER and the Golgi, and they function to move proteins between these two secretory organelles. And disruption of this pathway, again, leads to kind of proteinopathies in a number of tissues, such as the kidney, the eye, and the brain. So really, our central question is what factors and genes control the efficient and quality production of proteins, and can this teach us something about the pathogenesis of proteinopathies? So in order to do this, we kind of, again, as JT alluded, we took an approach developed by the Blaney lab called optical pooled screening, where we can take an epithelial cell line that is expressing Cas9. We can infect it with guide RNAs that disrupt or knock out the entire genome. We can subsequently sequence these guide RNAs to demultiplex which cells got which knockouts. And then we can subsequently integrate this with iterative imaging to basically mark or track both our secretory organelles as well as nodal proteins associated with the development of proteinopathies. So in order to do this at such a large scale, we made some key changes to the initial protocol developed by the Blaney lab that kind of allow us to scale up. So we optimized our optical pooled screening approach for compatibility with all commercial high-content screening automated microscopes that really can empower the next generation of optical pooled screening. We also increased throughput by integrating the protocol with automated plate loading and handling systems that, again, can empower drug screening and kind of and so on. And additionally, we can compress the screen by increasing surface area that we screen by up to 30%. And all these things together allow us to scale up pooled screening for therapeutic discovery. So just a kind of a brief kind of pause here. This doesn't happen in a vacuum. There's a lot of people to thank for this work. This project was co-led by a really talented BBS PhD candidate, Brianna Silverman, and a research associate, Jennifer Chen, as long as collaborations with CDOT, the GPP platform here at the Broad, Genomics platform, and then a lot of collaboration and advising from both JT's lab and Paul Blaney's lab. So just a kind of a quick aside on how we do this in situ sequencing and kind of some of the quality of this new platform. So as JT alluded, in situ sequencing of guide RNAs is a cyclical process where we read out the bases one by one. You can kind of appreciate over 15 cycles of in situ sequencing. We can kind of demultiplex an individual guide RNA using two-channel chemistry, which is kind of an evolution from the original protocol. If we compare these in situ read out guide RNAs compared to our kind of ground truth, our next generation sequencing data, which is on our x-axis, and then our in situ sequencing results on the y-axis, you can see a high correlation between our guide RNA distribution, suggesting that we're accurately reading out our guide RNAs. Similarly, if we look at our overall library skew, we see high concordance between our next generation sequencing results, our in situ sequencing results, and then our guide RNA library input. When we look at overall distribution of number of guides we're capturing per gene, we're consistent with other genome-wide screens where we're seeing around 1,000 cells knocked out per gene. So, kind of diving into the meat of the story, how are we changing the secretory pathway, and which genes and factors control the processing of proteins? So, using this kind of iterative and kind of massively paralleled approach, we can actually track the entire secretory pathway across the entire genome. So we can iteratively stain organelles starting from the ER all the way to the lysosome, as well as these nodal TMED proteins that are keynotes in the development of proteinopathies. So if we kind of delve into the data and we look at specific effectors of the secretory pathway, we can observe that positive controls are our top regulated downregulated guide RNAs for key organelle markers. So, for example, on the x-axis here, I show trans-Golgi contrast, and you can observe that the top downregulated guide RNA is the guide targeting the trans-Golgi. We can visualize this in individual cells, and again, you can see cells expressing guides targeting trans-Golgi 2 have an absence of a trans-Golgi signal. If we do pathway analysis of the genes of the guide RNAs that are downregulated, that downregulated trans-Golgi signal, they enrich for pathways that are essential for Golgi function, such as ER to Golgi transport, retrograde transport, and Golgi organization. So again, just highlighting the quality of the data. And because this is cyclical, often people ask, can we reproduce this data across different imaging cycles? So if we look in cycle 3, for example, and we look at the COP2 vesicle marker Sec13, we can see that our top downregulated guide RNAs encode for both Sec13 and Sec31A, while we see knocking out, for example, TMED proteins which live in this space, actually we see compensation of Sec13, which is kind of a nice positive control. Again, you can observe this at the kind of the individual cell level. And then lastly, again, we can go all the way to our final cycle, our sixth cycle of iterative imaging. And again, you can kind of observe the same thing where knocking out calnexin, which is our marker for the ER, is the topmost downregulated guide RNA. So these are all great positive controls. Again, if you look at upregulated guide RNAs, again, it's kind of the who's who of things that regulate the ER. So what else can we learn from this data, and how can we connect it to kind of the understanding of proteinopathies? So we can kind of start to understand how organelles work together and how this process, again, how organelles work together and how disrupting part of the signaling between organelles can regulate the secretory pathway. So kind of a classic example is the interaction between lysosomes and autophagosomes and the formation of autolysosomes. So if we kind of create a dot plot of things that change P62 or autophagosome intensity and LAMP2 or lysosome intensity, again, we can observe that knocking out the gene encoding P62 is our top downregulated guide RNA for P62 and LAMP2 for LAMP2. And then we can observe, again, we can observe shared mechanisms of regulation where inhibiting the VATPA complex or knocking it down can both activate P62 levels and lysosomal levels, and this is kind of concordant with the small molecule inhibitor bafalomycin. Similarly, we can observe unique regulators that specifically target one organelle and should have no effect on the other. So kind of our one nice example of this is our regulators of lipid transfer from the ER to nascent autophagosomes. So by knocking down members of this pathway, we can activate autophagy while not changing lysosomal intensity. So if we zoom out a little bit and we look at multi-organelle disruptors, so genes that disrupt multiple parts of this process, we can actually start to cluster genes into known pathways of biological function. So for example, we can observe organelles or guides that disrupt multiple organelles and that encode for, again, the transport of proteins early in the secretory pathway between the ER and the Golgi. Similarly, we can observe clusters encoding for genes encoding for transcriptional regulators, as well as ER stress or ERAD pathways, suggesting that, again, there is these shared nodes of regulation of this pathway that are important in the regulation of protein transport. So if we take a really big zoom out and we think of the 3,000 causative genes associated with monogenic rare diseases, and we kind of analyze which genes disrupt this pathway, something is pretty remarkable we find in the fact that almost 15%, 15% of these genes actually in our kidney cells result in disrupted organellar morphology, suggesting that these genes really, loss of function of these genes truly can disrupt this secretory pathway, and this can be a kind of a shared node for therapeutic discovery. So I'm just going to share with you one kind of case study and how we can use this approach to understand nodal mechanisms of proteinopathy pathobiology. So previous work from our lab has shown that TMEDs are kind of a known process by which misfolded proteins are entrapped in the early secretory pathway. These TMED proteins exist in co-regulated homoligomeric and heteroligomeric protein complexes. And if we look at the expression of for example, TMED9, one of the members of this complex, you can see that its expression is codependent with other members of this family. If we can recapitulate that in our screen, where we can, when we look at TMED9 protein levels, by knocking out TMED9 or TMED10, we can recapitulate what we see biochemically. Similarly, at the whole genome level, we can observe by knocking out TMED 10, 9, 2, and 7, we can observe a significant decrease in TMED 9 levels. And this complex family are highly dependent across the board. They exhibit both codependency and compensation for one another. So again, this is a nice kind of model where we can identify novel regulators for therapeutic discovery that are potentially specific to one complex. So, for example, we can look at TMED1 levels in cycle 3. You can see that TMED1, knocking out TMED1 biochemically, as well as knocking out TMED9 and TMED10, reduce protein levels, and we can kind of observe that in our screen, versus when we knock out TMED7, we actually see this huge compensation of TMED1. So again, further suggesting that there's kind of co-regulation of this protein family. So when we put all of our data together, we can observe kind of overall complex regulators that we can potentially target to more selectively target this nodal pathway. If we just look at just non-selective TMED complex regulators, it's again what we would expect in terms of things that alter protein transport, ER to Golgi transport, and so on. And then I'll just leave on this one point that we can start to now use this data to identify kind of more specific genomic targets that can be therapeutically relevant. So previous work from our lab has shown that removal of TMED9, a small molecule that removes TMED9 can kind of resolve several proteinopathies in the eye and the brain and the kidney. Now we can start to use this data to target more specifically potentially upstream or downstream in this pathway at the genome-wide level. So I just want to thank, again, our lab, my co-lead on the project, Brianna Silverman, and then our collaborations with CDOT, Genomics Perturbation Platform, and then Lattice Cures and Broad Auditorium for funding the project. So thank you.
Okay, thanks so much. I'd like to ask our speakers to come up. So Gabe, JT, and Matt. In the interest of time, we're going to close the session here, but if you want to come ask questions, we'll have a little rat pack of nerds right here, so come up and talk. Please come back at 11:15 to kick off our next session. Thanks so much.